we had such a nice session here, workshop. I think it was long due. What I'm going to present now is about recognizing a subgroup of colorectal cancer tumors, which is defined only by gene expression signature. But this time we are looking at the whole slide images and try to see how well we can perform in detecting these kind of special cases of colorectal cancer tumors. So first of all, some acknowledgements. But basically the data comes out from the PETA-3 trial, which was a long 15 years ago, a trial on comparing two different treatments. It was a negative trial, but the data was actually nicely collected, and then we could work a lot of other things more interesting than the trial itself, I would say. So let's consider first the case, let's say, in which we are all working. Just as a warning, I'm a computer scientist. I worked for a long time in computer vision, pattern recognition, and then I moved for the last decade in bioinformatics and biostatistics, and now I'm just going back a little bit to the origins, so I'm trying to combine things and to make a little bit new things around it. So let's consider that the case in which you start with a biological sample, and usually you have a pathologist and so which analyzes the sample and gives you a nice summary, qualitative data, which includes the histology, morphology, staging, and staining, and percentage of the tumor. But on the other hand, you have some extraction of DNA or RNA, and you apply a number of omics techniques. And at the end of the day, you end up with some quantitative variables, some continuous variables. You put them in a machinery, and you try to make up some stories around it. So, for example, we develop some signatures that are able to stratify the colorectal cancer patient population in a number of subgroups, depending on their survival. So that's, I would say, the typical workflow for a bioinformatician, for a biostatistician, does the biomarker discovery. Now, if you consider a little bit the case, how we extract from where the data comes from. So basically, you have a tumor. This is a particular case of a breast cancer tumor, but doesn't matter now. You have some regions, tumor regions, that mostly contain tumor cells, but also contain other cells, some epithelial cells, fat cells, and so on, from which the DNA and RNA is extracted. So all the cells are basically done kind of through a mixer, and you do the extraction, and then you do the hybridization of a Affymetrix chip, and then you end up with a matrix of data for the molecular data. So you have a matrix with, of course, the samples, the genes, and basically you have something like an order of tens of thousands of genes, and the order of, if you're lucky, hundreds of samples, maybe thousands of samples. Now the question is, how can we relate the expression for a particular sample and for a particular gene to actually the phenotype, or let's say the tissue where it comes from? So since of the mixing here of the signals and the cells, basically we cannot make a direct relationship. So you can see that this signal is kind of a weighted signal over the whole region of the cells here. So it's a kind of not well-defined problem, and in which we try to put in correspondence a number of genes in order of tens of thousands with the millions or billions of pixels somehow. So I will present quickly a little bit of background, a framework we are using for dealing with these kind of problems. And I just mention some other applications. So, for example, in breast cancer, we use the framework that I will present briefly, and we found some kind of patterns, tissue patterns that are correlated with gene expression. And this, some of those patterns can also be used to improve on the genomic score. So we have a genomic score or an image-based score, but when we combine the two, we actually have an even better discrimination of high risk versus low risk breast cancers. We also apply this for example, for predicting molecular subtypes of colorectal cancer. So molecular subtypes have been derived purely from gene expression data. And then we ask the question, can we find in a completely pathologist agnostic way patterns that are related to these subgroups? And actually it turned out that our patterns that we derived in computer vision, let's say, framework were quite significant and could be linked to the patterns that the pathologist also found on the same data. So we could identify papillary, also trabecular, and so on, architectural construction of the tissue. Now, a little bit of the framework that we are using. So the main idea is, since we don't have access to readily access to a pathologist to ask him always the questions, we try to go the other way around. We find some results, and then as a pathologist, they make sense. Because his time is quite valuable, and it's difficult to get him always. So what we use is to use local descriptors. We try to process the images on patches, extract some local descriptors that ideally would be scale and rotation invariant, and then try to aggregate them over the slides and find correlations with the gene expression or with the outcome, for example, the survival of the patients. So we use a kind of a whole slide coding, and the simplest, for example, is a histogram. Others can be graph-based or higher order statistics and so on. We have a lot of fun examples. So something that has been used quite intensively for the last decade or even more, it's the concept of bag of features, which is mostly used in image retrieval applications, but can be also used in the image recognition part. And basically it constructs a sparse representation of the image, and then it's used in the categorization of the image. So basically you construct a code book, summary representation of the whole set of images, and based on this code book, you recode your image in terms of a histogram of frequencies of, let's say, appearance of features from words from the code book. So in our case, for example, we started with an image, a whole slide image, in which, depending on how you see the question, you have the markings which are drawn on the slide. It's a good thing. It's easy to follow them. It's not such a good thing because the invariant prompt, for example, it's completely destroyed by the markings. But anyway, we could segment hand by hand this time just the interesting part of the tumor, which is just magnified here, and on this we did a processing. What is worth keeping in mind is that we don't know what we are looking for. So for this particular subgroup of colorectal cancer, there is no literature, let's say pathology literature, that could describe the features that we would be interested in. So we don't know if there are features that are correlated with this phenotype, with this genotype, let's say, and we don't know where they are located. So it's not easy to ask a pathologist the question, what should I look for? The next steps would be, so after the extraction of the region, we do the color deconvolutions simply to extract the hematoxylin, let's say, signal, on which we are applying a Gaussian filtering, and then we do the standard code book construction, which consists of extracting the local descriptors and learning the diction. And of course, at the end, we have to do the recoding of the image. So what it means, the code book construction means that we extract patches from the image. These patches are described in terms of some local descriptors, like wavelets, like local binary patterns, HAR, whatever you can come up with. We do a simple k-means clustering, so it's a vector quantization problem, and then we extract a number of representative, let's say, code books, which are the centers of the clusters, the k clusters that come out from the cluster. So there are a number of questions that you have to ask when designing your system. What would be your local descriptors? What would be the size of the local neighborhood? What would be the sampling strategy? Do I look for every position? Do I look only for non-overlapping positions? Do I look for random positions? So there are different sampling strategies that you can think about. For this particular application, we took out the SURF descriptor, so it's a speed-up robust features. Basically, it combines a point of interest detector, so it detects, let's say, points of high gradient in the image, and with a local descriptor that is multi-resolution and rotation invariant. So it has a number of advantages over the other descriptors. But it has also a drawback in the sense that low gradient images, so those that are constant, close to constant gradient, they are not detected. So, for example, the stroma, which is kind of a uniform pattern over the image, is not so interesting from its point of view. So it is not taken into account in this application. And actually, sorry, you can also see here the points where like neighborhoods are extracted in some regions that contain high gradient, high variability of the image, and not in those that are more constant, which contain fat and strong. So that's something that we have to deal with in this part. Another question to ask is, what would be the code size, or the codebook size? So should I fix a priori? Somebody would say 100 clusters in the code would be enough. Should we try to optimize it over the image and so on? So we can use also this kind of gap statistic, which is a way of testing if the size of a cluster is optimal or not. Or we can use simply, let's say, another classifier or a simple classifier just to see when it achieves the peak performance, so in a cross-validation group. So what I was looking at is a simple classifier, DLE, so diagonal linear discriminant, and I just picked up the position rate suggested to be in the inner cross-validation loop, which suggested to be the highest performance. Another, so once you fix these parameters, meta-parameters of your problem, you do the recoding of the image, and the recoding means basically for each of the code books, code words of the code book, you extract, you count how many times you have features of similar appearance. So you end up, for the whole image, you end up with a histogram of features. Once, so clearly this is a drastic reduction in dimensionality, so it's only the coarse, the very coarse appearance of the image that you can extract. So it's nothing to have the finest detail that a pathologist will talk about, for example, when looking at the image. On the other hand, it's only the most dominant patterns that are surviving of this, let's say, recoding step. And in the sense, it's similar to what it happens also in gene expression, where only the strongest signals, actually the most dominant types of cells, will give you a signal to work on. So there are some similarities, if you want, with the gene expression data. We also tried out let's say, modification of this representation in the sense that we don't use only the K code words from the dictionary, but we introduce a pseudo code word into which all the patches that cannot be reliably assigned to one of those K are assigned. So something that I'm not sure where to put, I'm just putting aside, just to see what, just to improve, let's say, the specificity of these K words. So what we end up with, it's a number of 50 features. As you can see, they are in different, let's say, patterns from the tissue, and they have also different sizes. As I said, it's a multi-resolution approach. And they are located mostly in the high, let's say, density, tumor density of the tissue. So this part, for example, is less populated. But, so here also the image, the color of the patches codes for the index, let's say, of the code. So you see a little bit where the code books are distributed over the image. And you apply this to recognize the so-called BRAF-like tumors. So what are the BRAF-likes? And when you have a population of colorectal cancer patients, most kind of 8% to 10% of those patients will harbor the BRAF mutation, the V600E mutation. Now, some years ago, we tried to predict, to learn, let's say, what would be the patterns, gene expression patterns characteristic of these mutations, and we found that actually another subgroup of patients not harboring the mutation, they express the same pattern. So this is what we call the BRAF-likes. From a pure, let's say, machine learning point of view, they are false positives when you try to classify. But actually, they have a very nice, let's say, they display a very nice expression pattern, and more importantly, but from a diagnostic and prognostic point of view, they have a survival experience very close to the true BRAF. So they have high risk of relapse and high risk of death after relapse. So that's why they are a relevant subgroup of patients from a whole population of colorectal cancers. But it's a group that is not well recognized. So if you want to recognize it, you have to resort to 64 panel of genes that you have to provide and so on. The question is, can a pathologist recognize earlier on the subgroup of patients actually to test data? So that's why we train the classifier, and we use 113 samples. They're all magnified stable and stage 3 colorectal cancer, so there's a more homogeneous subgroup of patients. And from this, we divide in 2 sets, and 40 samples we use for training, 73 for testing. And we use only equivalent of 2.5 magnification of the whole slide. So the ground truth is given by a gene expression signature, which again cannot be seen as an absolute truth. It's always a little bit fuzzy. The code, yeah. Sorry.
So did you also include the real BRAFs, just BRAF?
No, just the BRAF, the real BRAF are not there.
So there's wild type and BRAF.
Yeah. So the optimal code size was 50 for us. And we found 9 code books that are actually associated with the BRAF mutant post-hoc, let's say, analysis, that are also the BRAF mutant status. But we also found 10 code blocks, for example, which are associated with the Mishima status. So clearly, these patterns are picking up also some characteristics for the morphological aspects of the gene. With the 30 features at the end of the LDA achieved almost 92% confidence, 92% accuracy, with only 6 misclassified out of 73. So the sensitivity is slightly better than the specificity, meaning that most of the high-risk patients are recognized. Of course, we can tune a little bit the thresholds, and we can improve the sensitivity or the specificity, depending on if you're working on a filtering, let's say, scenario or in a confirmatory scenario. But then when we put these predictions into a Cox model, we've seen that actually we are also able to predict to make some prognosis out of these features. So if you compare, this is the molecular signature. Of course, the figure is significant. We have almost 2.2, I guess, hazard ratios here. But without anything else, without tuning any threshold, we will achieve almost significant also prognostic with the image features. So the difference between the two is 6 patients that fall out in the wrong category. Of course, the sample size is too small, let's say, to draw any conclusions, but at least it's a promising, let's say, test. And as pointed out in the previous presentation, you can run it overnight and have the next day some, let's say, hints about your cases. So conclusions. The histopathology images can bring new clues to the bioinformatic analysis. The most important part is the feature extraction for us. And it's also an interesting approach to derive new biomarkers, so they can also be proxy biomarkers for gene expression biomarkers already existing. And of course, the future is to explore new and various classifiers to see new features and so on, to see how can we improve on these results. And I guess that's all. This is something that I found on the web. Don't be scared about it.